Good evening. Good evening and welcome to the 2020 Forbes Lecturer. I'm Catherine Carr, the president of the Grass Foundation. And I'd like to welcome you all to the Forbes Lecture. Before I hand over to my colleague for the introductions, I have one pleasurable announcement to make. If I could have the first slide. No, there we go. Last year, the Grass Foundation decided to institute a prize to recognize especially talented people from minority groups. And we had many very wonderful candidates. But our winner of the Henry Grass Rising Stars Award for 2020 is Dr. Nancy Padilla-Coriano, who was a postdoc at the Salk and has now moved to the University of Florida. She's in the audience. Would you be so kind as to stand up? In other years, Nancy would be giving a lecture in one of the courses, but I urge you to seek her out. She's done really outstanding work. Now I'm going to hand over to my colleague, Dr. Andre Fenton, who's going to introduce our lecturer. Andre is a former course director of Neural Systems and Behavior, a Grass trustee, and a professor of neuroscience at NYU. Andre.
Good evening. Can you all hear me? Thank you. Welcome to tonight's MBL Friday evening lecture. My name's Andre Fenton. I'm a professor of neuroscience at NYU. I'm also a MBL Whitman Center Fellow this year. I'm a former co-director of NSMB, as you recently heard, and a big fan of the MBL. I'm going to ask your indulgence as I have a number of stories to tell that will hopefully put tonight's events and lecture into historical context. I want to thank those of you who have come to the new Cornelia Clapp Auditorium, and those of you who are joining us online, virtually. So this summer, the MBL is recognizing past members of the community who have made extraordinary contributions to this storied institution. So it's my privilege this evening to share with you highlights of the life and work of Stephen Kuffler. He's known as the father of modern neuroscience, and you'll understand why. He was a faculty member here, an investigator, a trustee, and a corporation member here at MBL. Most knew him as Steve, but he was born in 1913 as Willem Kuffler. And he didn't adopt his new name, Steve, for over 20 years. Steve spent the first 10 years of his life, from what I can tell idyllically, on his family's estate in Hungary, before being sent to Jesuit boarding school in Austria. He eventually chose to pursue medicine because of its international character, and he was not to be disappointed. During the 5 years it took him to earn a medical degree in Vienna, he spent 6 months in London and trekked through the Middle East and Egypt to evade the turmoil of the receding Austrian Anschluss. The Germans ultimately invaded Austria in 1938 during Steve's clinical residency. His paternal grandmother was Jewish, and the Nazi hostility was patently obvious. It was especially so when he had to perform a postmortem on a colleague who had been murdered by the Nazis. Steve fled to London and soon after that to Sydney, Australia. In Sydney, Steve gained his footing in experimental science, or rather unexpected ground, the tennis court. He was volleying with neurophysiologist and future Nobel laureate, the yet unknighted John Eccles. Eccles hired Kuffler as an assistant in his physiology lab at Sydney Hospital. Eccles was using cats to study the neuromuscular junction. That's where neurons interface with muscles to control those muscles, to activate them. And Kuffler was joined there by future Nobel laureate Bernard Katz. Katz convinced him to take advantage of simpler non-mammalian species and systems like frogs. Because of this encouragement, it seems, Steve was, and his talents, his special hands, he was the first to master a dissection that allowed him to isolate a single nerve fiber and the muscle that it innervated. This feat drew international kudos, and it helped earn him a fellowship at the University of Chicago. Shortly thereafter, he accepted a professorship at Johns Hopkins University School of Medicine. His research career took off. First came some seminal studies on skeletal muscle fibers and muscle spindles, then experiments on the mammalian retina and the stretch receptors in those muscles, in particular in crustaceans. Some of these experiments were actually conducted here at MBL, where he began spending the summers in 1947. In 1959, Kuffler relocated his team from Hopkins to the Department of Pharmacology at Harvard Medical School. Within a decade, less than a decade of being at Harvard, he became the inaugural chair of their Department of Neurobiology. This is quite remarkable because this is the first neuroscience department in the world. Unlike today, in 1966, the notion of uniting experts from a variety of disciplines to study the nervous system, that was totally unprecedented. Steve also established the first experimental classes that were centered on the nervous system at the MBL. These were taught by Dave Potter and Ed Fershpen. They were known as the Nerve Muscle Program, and these classes preceded MBL's neurobiology and the neural systems and behavior courses, the latter's the one I co-directed. Kuffler's program drew disciples of many disciplines, and he was instrumental in introducing them to the budding field of neurobiology. Steve was known for selecting just the right organism for each research question. As with his choice of crustaceans, while working to pinpoint the role of a nervous system chemical called GABA. Crayfish and lobsters were optimal subjects because they contain this inhibitory as well as excitatory nerve fibers that were large in diameter, so easy to access, and few in number. Thanks to that intuition, that elegant simplicity of the system, Ed Kravitz, a former co-director of MBL's neurobiology course, and his group with Kuffler and Kuffler's strong support, they demonstrated that the inhibitory, but not the excitatory fibers released GABA. This indicated that GABA was an inhibitory neurotransmitter. And back then, only 2 other neurotransmitters had been identified, and GABA was the first inhibitory one. This was big news. After Steve stopped doing experiments on the GABA system, guess what? He transitioned to studying the function of glial cells in the nervous system, and he introduced the use of the leech for those studies. Steve was accustomed to defending his groundbreaking work, and one of his favorite forums, I'm told, was MBL's Monday night seminar series, colloquially known as Fight Club. I'm told here the research titans would discuss, more often debate, their scientific theories. Kuffler returned to Woods Hole nearly every summer with his wife, Phyllis, and children. The Kuffler home near Stony Beach was known as a welcoming place, filled with lively conversation. Phyllis ran a symphony, I learned, inaugurated a symphony in Woods Hole, and was an early organizer of the MBL Club and their activities, which to this day promotes the activities for MBL families that enable us to also enrich our children. Steve died in 1980 and was buried at the Church of the Messiah here in Woods Hole. We're honored to have Steve's children, Damien and Susan, here at tonight's lecture. And we have online Lauren in Maine. Welcome. We're pleased to honor the life and contributions of Stephen Kuffler, a pioneer in neuroscience really as a discipline, and an eternally beloved member of this MBL community. And now I'd like to introduce tonight's lecture. Thank you. I have a tremendous honor and privilege this evening of introducing the Forbes Lecture for 2021. Each summer, the Grass Foundation brings a world-renowned neuroscientist to MBL. This year, we are pleased to welcome Professor Guoping Feng, the Poitras Chair, Professor of Neuroscience at MIT's McGovern Institute for Brain Research. Before I introduce Guoping, I'd like to tell you a little bit about the Grass Foundation and about Alexander Forbes. After all, this is the Forbes Lecture Series. Next slide, please. Albert and Ellen Grass were truly wonderful and generous people. Albert and Ellen first met in 1935 while in the Department of Physiology at Harvard Medical School, and they were married the year after. Albert, who had an engineering degree, he spearheaded the design and the fabrication of the first multi-channel EEG machine in 1935. Since then, as I think many of you know, EEG machines have played truly an indispensable role in the diagnosis and management of epilepsy and other brain disorders. Ellen received her AB and master degrees at Radcliffe. She was a PhD student at Harvard, and there she studied the electrophysiology of the auditory cortex. After their marriage, Ellen left graduate school to work with Albert to found and direct the Grass Instrument Company. The success of the company resulted from what I can tell, powerful combination of Albert's expert engineering and Ellen's scientific expertise. From the very beginning, Albert and Ellen Grass were committed to supporting young neuroscientists in their nascent early stages of their careers, both here and abroad. They were incredibly generous. With the financial success of the Grass Instrument Company, Albert and Ellen put their commitment into action. In today's parlance, they walk the talk. They established the Grass Foundation in 1955. In the 66 years since its establishment, the foundation has supported a variety of activities, mainly focused on facilitating the careers of young scientists. These activities provide support for the neurobiology course, for the neural systems and behavior course here at MBL, as well as for the flagship Grass Fellows Program. And that program provides a truly extraordinary independent research opportunity to help young scientists figure out and establish what their research interests might be, as well as their careers. So I'd like to now tell you a little bit about Alexander Forbes. Next slide, please. Alexander Forbes was a founding trustee of the Grass Foundation. He was a close associate of Albert and Ellen. He was an MBL scientist and the first Forbes lecturer in 1959. He was one of the founders of modern neurophysiology. He's credited with building and using the first vacuum tube amplifiers for neurophysiological experiments. Before this pioneering achievement, recording the bioelectricity activity of neurons was performed using string galvanometers. Next slide. Thank you. The Forbes family has many well-known members. For example, John Forbes Kerry, John Kerry, former senator from Massachusetts, Secretary of State in the Obama administration, and he was recently named the first United States Special Presidential Envoy for Climate by President Biden. Alexander Forbes was himself a grandson of Ralph Waldo Emerson and a son of the first president of Bell Telephone. How can you go wrong? He was educated entirely at Harvard, where he received his medical degree in 1910. After receiving his MD, Alexander worked in England with the 1932 Nobel laureates, Sir Charles Sherrington and Lord Adrian. You can see Alexander here with the laureates in Ipswich. This picture was taken in 1938. Soon after finishing medical school, Alexander made the first electrical recordings of nerve action potentials during the flexor reflex back in 1915, using string galvanometers with his colleague Alan Gregg. So Forbes met Albert Grass in the 30s. Albert had joined the group to build a better EEG recording device, and this marks the beginning of the Grass EEG ink writers and of the Grass Instrument Company. Forbes was a fixture at Woods Hole, not just because of his affinity for the MBL. The Forbes family owns Nashan Island and most of the Elizabeth Islands, just a short boat ride away. So I'd like to now move from our celebrated past to our celebrated colleague, Professor Feng, who will tell us about the extraordinary potential of our futures. Next slide, please. I really am truly very pleased to welcome the 2021 Forbes Lecturer, Professor Guoping Feng. Welcome, I should say, welcome back to MBL and Woods Hole. Guoping is the Poitras Chair, Professor of Neuroscience, and the Associate Director of the McGovern Institute for Brain Research at MIT. He's a member of the MIT Harvard Broad Institute and has a number of leadership roles. He's the Director of the Model Systems and Neurobiology within the Stanley Center for Psychiatric Research. Professor Feng's research is truly breathtaking. He investigates the development and the function of synapses. Those are the communication connections between neurons. And he uses and investigates the molecules that make those synapses. This is the stuff that realizes all of the operations of those connections, those synapses, and the neural circuits that they define. Guoping's research is at once bold and impactful, but I have to say, in my review, it's also beautiful. After Mao's Cultural Revolution, Guoping was amongst the early cohort to be allowed a university education based on his high test scores. In 1978, he was assigned to attend medical school at the Department of Medicine at Zhejiang Medical University. I believe he wanted to be an engineer, but in 1978 China, that was not up to him. Guoping then earned a master's degree in pharmacology at the Medical University in 1985. From what I can tell, during that time, this is a conjecture, Guoping became keenly aware of the immense suffering that brain disorders cause, and frankly, the blatant inadequacies of our understanding of them. And that's where I believe he started to develop a vision of how we could do better. In 1989, he began his PhD training in molecular genetics, working in fruit flies, specifically Drosophila genetics, with Linda Hall at the State University of New York at Buffalo. He graduated in 1995, and for his thesis, he cloned a gene for one of the proteins that makes the pore of the voltage-gated sodium channel that's responsible, or one of them that's responsible for the electrical activity in Drosophila. Can you go to the next slide, please? Guoping is not a stranger to MBL. He was a Grass Fellow in 1994. Here is Grass Fellow Feng, sitting on the corner to your left. Next slide, please. 2 years later, as a recently minted PhD, Guoping was the Associate Director for the Grass Lab here in MBL. I want you to take note, if you can see it, of the statement on his t-shirt. Life is good. Apparently, he's ever optimistic. The year before, Guoping began his postdoctoral training with Joshua Saines. At that time, it was in Washington University in St. Louis. It was there that he diligently and painstakingly figured out how to genetically modify specific neuron types in mice so that they would express a fluorescent protein of different colors derived from green fluorescent protein, which was isolated from jellyfish by Professor Osamu Shimomura here at MBL. Professor Shimomura shared the 2008 Nobel Prize for this work. In a sense, Guoping became an engineer and so much more. That engineering has allowed the field of neuroscience to see specific cells and their connections. I counted 36 mouse strains from his efforts in the Jackson laboratory catalog. And that has enabled hundreds of laboratories around the world to study nervous system circuits. Next slide, please. Guoping joined the Department of Neurobiology at Duke University in 2000 as an assistant professor. In 2007, he identified that the mutations of a synaptic protein in mice causes those mice to develop obsessive grooming behavior. And this resembles symptoms of obsessive compulsive disorder in people. He joined the MIT faculty in 2010 and his contributions accelerated. He studies the molecular basis of a number of disorders, including autism, schizophrenia, ADHD, and feeding behaviors. His contributions are many and fundamental. He's pioneered the field's ability to do genetically precise circuit neuroscience in mice and in non-human primates, all in pursuit of a better understanding and better treatment options for human disorders. He's published at least 147 papers, and his work has been cited by others over 27,000 times. His work has been recognized by numerous awards and tremendously successful funding from governments, private foundations, and companies. From the page of his awards in his CV, I'll mention only 3. He was elected a member of the American Academy of Arts and Sciences. He received the Scientific Innovations Award from the Brain Research Foundation, and last year was recognized for his mentoring by the Frank E. Perkins Award for excellence in graduate advising at MIT. I'm going to close with a quote from his postdoctoral advisor, who's now a colleague, and former GRASS trustee, Joshua Saines. Guoping is sort of infinitely optimistic and infinitely energetic. I'm confident we're in for a treat. Professor Feng's 2021 Forbes lecture title is The Future of Brain Disorder Treatment, Breakthrough Technologies and Ethics. Please join me in welcoming Professor Feng to the podium. Thank you. On behalf of the GRASS trustees, I would like to present you with, I'll open it,
Thank you. Oh, wow.
memorial medallion.
Thank you.
Thank you.
Thank you. Now the test start. Can I make it work? All right. Thank you, Andrew, for a wonderful and generous introduction. So it's a great honor to be here to give a Forbes lecture. It's a great honor for anyone to give a Forbes lecture. But it's a special honor for me because I was a GRASS fellow, and the GRASS fellow experience literally changed my life and changed my family's life and actually changed my, defined my career. So for that, before I give a talk, I want to give some special thanks, especially the GRASS family. I had the honor to meet Ellen Grass many times, visited her home at Braintree, and she's a wonderful, wonderful and visionary woman. I also want to thank GRASS Fellowship Program. When I was a fellow, Steve Zotterly was the director. And after that, He became my life coach and also lifesaver. For example, today I forgot bring a tie. He said, "Oh, I have two in my pocket." So whenever you need, he has it somewhere in his pocket. So, and I also want to thank trustees, GRASS trustees, for their dedication to nurturing young scientists. So it's an unbelievable experience to be a GRASS fellow. And science discovery is hard. Sometimes it's frustrating, sometimes stressful. I was the final year of my PhD, and if you're a student, you understand. If you are not there yet, you will get there. So it's very stressful. And but when you get here with the GRASS support, not only you, but your family for a summer here, you realize that there are so many people dedicated their wealth, their time, their effort, and their love to train, nurturing young scientists. So make you feel there's nothing you cannot do. There's no reason you should not work hard to make scientific discoveries and to benefit the society. So that literally changed my life and changed my approach and philosophy, what kind of research I want to do. And I also want to thank Marine Biology Lab. MBL is a special place. And in most places, you work really hard, you have all kind of politics, everything. But here, MBL really dedicated to explorative and collaborative and fun science. Fun is the part I experienced fun doing science first time was at MBL. So I'm very grateful. And I also want to thank the whole community. It's incredibly friendly and welcome community. So for that, I will be forever grateful. And I'm really honored to give a Forbes lecture today. Thank you. So, thank you. So today I want to talk about the futures of brain disorder treatment. And you all know brain disorder actually is the most debilitating leading cause of disability. This is the data from World Health Organization. The reason they are leading cause of disability is because, I'm trying to see what I can find at the point. Okay, so the reason is because for many of the for example, for psychiatric disorders, many of them are onset times are at your best productive years, right? From teenagers to to the young adults. So this red part is mental health, mental illness. You can see that the majority of them onset are very early, the most productive years. So, and together with neurological disorder, thank you. With neurological disorders, actually this is the leading cause of disability. Now, why, they become a leading cause? Many other high incidence disease, but they have medicine. For brain disorders, we have very few treatment options. I use this data, this actually borrowed at Skolnick, who was the founding director of Stanley Center. So if you look at the drug development from a drug available, the number of mechanistically new drugs, not the Me Too drugs, from 1950 to, this is 2010. And if you look at the cardiovascular drugs for cardiovascular problems, you can see they are dramatically increased, the class of drugs. But for mental illness, for example, depression and schizophrenia, barely nudged. This is not because lack of effort. Hundreds of millions of dollars have been spent in developing drugs, and tens of thousands people have labs and people have studied brain disorders. But why we have so few successful stories? There are probably many reasons, but I list a few of them. The number one reason is we lack the understanding of causes of brain disorders. The second is that we lack the understanding of pathological mechanisms. Without understanding that, we cannot develop medicine. And the third is we lack adequate animal models, right? So cancer is a very difficult disease. But at least they have the advantage, whenever the tissue you need, you can get it from surgeon. They always want to cut them off. For brain, whether you study autism, depression, schizophrenia, we cannot get the live tissue. Occasionally, you may get it from surgery, but it's a very little piece for, let's say, epilepsy. But we know every little piece of brain is different. They have different cell types, different connection, different function. So that actually help only very little, right? So we heavily rely on animal models. But how can you tell an animal model is schizophrenic or bipolar or autistic? So these are the very difficult problem. The fourth thing is we don't have biomarkers, right? So if you have a cancer, you know what it's looking for. If you have leukemia, you take a blood, you can tell. But for psychiatric disorders, it's very difficult, right? So we used to say if you send a patient, psychiatric patient to 5 psychiatrists, you may come back with 10 different diagnoses, right? So that's a difficult thing. So the lack of biomarker also, make the drug testing and the therapeutic testing very difficult. So, but in the last 10, 15 years, we had a lot of progress. And now we are very optimistic that we will have breakthrough treatment in the near future. The number one reason is we had a lot of genetic advances, a lot of advances in genetics, in understanding genetic of psychiatric disorders. So with a very large scale genetic study, because of the reduced cost and the new technology, we now know that genetic mutations or genetic variations play a very important role in some of the disorders. For example, autism spectrum disorders and schizophrenia, but less in other disorders like depression and addiction. And there are also other factors too. It's not all genetic. There are epigenetic factors, environmental factors. We also know that by so far, most of the disorders are polygenic. That means more than one gene contributed to a particular disorder in a particular person. And however, we also have some monogenic causes. That means some of the patients are caused by a single gene mutation. Now that gives us a hope, because once we know which gene is causative, then we can start to study them. We can model them and study them. And one surprising finding is that although clinically we diagnose them bipolar, schizophrenia, and autism or ADHD, there are many of the risk genes are shared, are the same genes, it's just different mutations. And so the other thing which is surprising, now it's not surprising, but 20 years ago, when you asked me, I would be really surprised. There are hundreds of risk genes for each disorder. So with all this, how can we study them or develop treatment if each of the diseases have hundreds of risk genes? So scientists have been looking for, are there common pathways, right, converging? Because whether for example, autism spectrum disorder or schizophrenia, although there are some common features, but most are very distinct. One is very early onset, one is teenager or high school or early college. So large-scale data analysis suggests that at least there are a few pathways are common. One of the pathways is they converge to synapses. Many of the risk genes are important for coding proteins important for synaptic function. So this gives, and as Andrew mentioned, synapse is the apparatus that neurons transmit important for neuron-neuron communications. So this kind of pathway analysis and combined with genetic study, give an unprecedented opportunity for neuroscientists to now try to understand what is the mechanism that a genetic mutation can contribute or even cause brain disorder or psychiatric disorders. So my lab, like many other labs, study how these synapses develop and how they function and what will go wrong if the genes actually important for synapse is mutated in patient. So in human brain, we have 80 billion neurons about, and there are trillions of synapses. The difference, the unique feature of our brain, unlike a liver, every single cell in the liver, they can function autonomously, right? In the brain, they don't. They have to communicate with another neuron, with a bunch of other neurons, form a circuit, right? So if I raise my hand, there are many, many neurons is already activated and calculated, computed, then send a signal to the to the spinal cord, the motor neurons, then the motor neurons send a signal to a particular whole bunch of muscles. And if I do this, then this muscle has to contract and this has to relax. So all these are neural circuit problems. So the recent study I use autism spectrum disorder example, use large scale genetic study suggesting that many of the risk genes or causative genes are important for synapses, for synaptic function, for synaptic development, or for synaptic plasticity. So at the synapses, there are two parts. One is the neuron sending signals we call the presynaptic terminal. The other part is from the neurons to receive signal. Right? You transmit a signal from one neuron to another. My lab and many labs are very interested in how these postsynaptic side, we call this presynaptic, we call this postsynaptic, how these postsynaptic side assemble. These synapses are very small. This is about 2 microns. And in 100 nanometer area, we call the postsynaptic density, there are at least 1,500 type of proteins clustered there. So if you think about it, we say if there are 100 ways to build it, there are 100 ways to break it. Right? So it's not surprising that many of the genes important for synapse function are risk genes for brain disorders, including autism spectrum disorder and psychiatric disorders. Today, I'm going to focus on, so how do they assemble all these 1,500 proteins at the synapse? So they use one strategy is use a scaffold. You build a scaffold, then you bring everything else in and out dynamically. Our synapse, our brain function, always dynamic. So one of the scaffold is called the PS95 SAPAP Shank scaffold. They have many protein-protein interaction domains. So the former scaffold, they interact with the important receptors, ion channels here to receive signal. Then they interact with cytoskeleton. So anchor them here. They also dynamically regulated. So today I'm going to focus on one of the protein called SHANK3. The reason we focus on this is SHANK3 is a very important scaffolding protein for the synapse, and the SHANK3 mutation monogenically causes severe neurodevelopmental disorder and autism spectrum disorder. So you're probably very familiar with ASD now. There are 2 major symptoms. One is persistent deficit in social communication. The other is restrictive repetitive behavior. And these are all often associated with many other problems, including sensory abnormalities, seizures, ADHD, right? So SHANK3 mutation is a monogenic cause of severe ASD, and actually is one of the most common monogenic cause of ASD. It accounts about 2% of all severe ASDs with intellectual disability. And the symptoms are very severe. Their IQ on average is 30. So they will never be living independently. They always need someone to take care of them. And they have hypertonia, they are minimum verbal, and they have autism spectrum features. So now with a monogenic cause, you can generate animal models. We don't know whether animal will behave like autistic because there's no way to diagnose a mouse or any animals, because we barely, a lot of times we cannot even actually diagnose humans, right? So, but with genetic data, this mutation is exactly mutation cause autism spectrum disorder in humans. You can put the same mutation in animal to look at what is the consequence to the function of the synapse, to the circuits, and to the build-up of the nervous system. So allow us to really try to understand what is the cause of this mutation that will result in autism spectrum disorder. So we started with mouse models and generated a deletion in a mouse, and we can see indeed the SHANK3 is a scaffolding protein, because without the SHANK3 protein, you have molecular defect. Other scaffolding proteins are lost. Other scaffolding protein lost neurotransmitter receptors, which is critical for receiving the signal. And also, if you use electrophysiology to recording, we see a functional defect. Neuron-neuron communication is disrupted. And we also see that they have reduced the synapse numbers. Their structure changes, that neuron-neuron connectivity connection is reduced. All this lead to synaptic failure in the mouse brain, and you see a lot of behavior abnormalities. This work was done by two former graduate students, João Peca and Katia Feliciano. João is now a faculty assistant professor in Portugal, Coimbra University. And so these mice actually have many of the phenotypes we think it's relevant. We cannot say it's the same, but it's relevant to ASD. Right? So first of all, you have repetitive compulsive grooming behavior. They grooming their skin off, you can see sometimes. And they have social interaction deficit. Although mouse social is different from human, but they do social. So they have social interaction deficit. They have sensory oversensitivity. They have anxiety-like behavior. They have reduced the locomotion. And in humans, we have hypertonia in this patient, and they have impaired motor learning and other learnings. So for all these phenotypes, we try to figure out what are the neurobiological mechanisms that can lead to such diverse phenotype. Is there a common mechanism that we can find and we can develop a drug to treat them, to help these kids? So we start one by one. For example, I'm not going to go through all the details. I'm going to give you a summary. So for example, why are they grooming like this, right? It hurts them, but they keep grooming them. So it's a repetitive compulsive-like behavior. We found that actually this defect is one of the brain region called basal ganglia in the striatum. Striatum, in the basal ganglia, there are two pathways. Basal ganglia is critical for our motor, for habit formation. So, and for many other functions. And so we found that the defect is one group of cell, and it's called indirect pathway medium spine neurons. They have reduced the activity, so lead to overactivation of direct pathway medium spine neuron. And we think that's form a recurrent circuit activity and repetitive compulsive behavior. So if we, in the mutant animal, if we slightly increase the neuronal activity of this group of neurons to correct the defect without put the Shank3 back, you can actually eliminate rescue the repetitive grooming behavior. So now one thing is clear. So is a similar thing happen for other phenotypes? It turned out to be not. So these mice, as I mentioned, they have social interaction deficit. Instead of showing a bar graph, I think it's easier to show you a video. So this is, we put two mice together, strangers, into one cage. And this, on the left side is the Y type. On the right side are two mutant mice. You drop them together in Y-type mice, they usually will looking for each other and they will start sniffing the other mouse behind. That's typical mouse social. And you will see, but for the mutant mice, even they bump into each other, you will completely ignore. You will see very soon. These are Y-type on the left and mutant on the right, right? So you can see they bump into each other. They're not interested, and their head getting close, right? They will separate. And Y-type, you see, they will sniff other mouse behind. Then they like them, they start social, they cuddle and stay together, right? So I cannot say this has anything to do with the human social, but it's a very interesting biological phenomenon. Why a single gene mutation causes such different phenotypes? So my former postdoc, now a professor in China, continued to work from after left my lab, showed that this defect actually one of the region is the anterior cingulate cortex. These are, instead of in the basal ganglia, it's actually in the cortex. And the defect is actually reduced activity in pyramidal neuron, which these are projection excited neurons. So now you have a very different mechanism, different brain regions actually lead to a social interaction deficit. And besides the social interaction deficit, they might also have a sensory oversensitivity. Now I'm going to show you another video. In this video, they are mice are in their home cage. This time left side are mutant animals, right side are Y-type. We drop a plastic ball in the middle. This changes their, it's a new object. They usually mice with new things they like to explore. What you will see is that Y-type animals on the right side will start to explore. Right? They use whiskers very often, so you touch it, then they will go on. Mutant animal, you can see when their head getting close, right? They will suddenly startle back. So we think that when their whiskers touch it, and they are super sensitive, they are very afraid. This is what Y-type mice pretty soon will get on top of it. You will see, and they're playing with it. They will never want to touch it, right? So you can see we thought this might be a really good model for study sensory oversensitivity. You know, in autism patients, many of them have sensory abnormality. Some are very sensitive to light, to touch, to noise. Some are hypersensitive to light, touch, and other things. So we use the calcium imaging approach to look at what is wrong in the somatosensory cortex, which is connected to the processing the sensory information from peripherals. What we found is in this case is actually another imbalance between the excited neurons and inhibiting neurons. What happens is these excited neurons in the somatosensory cortex is instead of reduced activity, they are hyperactive. So that may be able to explain why these animals are hypersensitive to touch, right? This is the risk area. And the reason actually is not a defect directly from excited neuron. It's actually defect is from reduced activity inhibiting neuron that innervates this excited neuron. So we then made a conditional knockout, only knock out the SHANK3 locally in somatosensory cortex, in inhibiting neuron, you get the same results. You get excited neuron hyperactivity, you get sensory hypersensitivity. So now for the third phenotype, it's another different mechanism to explain a neurobiological mechanism underlying this kind of phenotype. So this work was done by two postdocs. Chen is a postdoc fellow in my lab, and Chris Deist is a postdoc with Chris Moore at Brown University, and Chris is an expert in somatosensory system. So now that we realize it's not as simple as we originally thought. We can find a common mechanism. It's a monogenic, one gene mutation. How complex it could be? It turned out to be really, really complex. So now lead us to, we have to think about if we want to develop treatment, how can we, what is the approach we can use, right? So this is just a single gene mutation, a monogenic cause. They are neurobiological defects in multiple cell types and multiple circuits and linked to various behavior abnormalities. And this is a single monogenic cause of autism. And we know there are hundreds of causative or risk genes for autism spectrum. How are you going to develop a drug that can correct them all, or even a fraction of them? So that leads us to really, sit down and really think about if we really want to develop a treatment, what approach we can use. So we saw at least the two things that can be in the near term, it will be beneficial. One is for monogenic causes with the new technology, the best way is probably the correct genetic mutation. Because scaffolding proteins, they interact with many, many other proteins. You can study one of them, you can fix one of the pathways, signaling pathways, but 10 or 20 others are still defective. But if you correct the genetic material mutation, then everything is corrected. That's at least a possibility, right? And so but monogenic disorder only a small fraction. In ASD, maybe 25%. In schizophrenia, we haven't found a monogenic one yet. And they are high-risk genes, but they may need other genetic variations or other factors. So how do we deal with that? So in SHANK3 study, we showed that every phenotype comes with a circuit defect. And we can figure it out as a neurobiologist, at least in animal models. If we figure out that, and for these polygenics, we can, by modulating circuit function in these polygenic cases, we can at least relieve some of the most debilitating symptoms. So I'm going to give a couple examples of this to illustrate how we might move forward to develop treatment. So use autism as an example. Autism is a neurodevelopmental disorder. Right? So one of the issues is if you say, I want to correct the genetic mutation, the first thing we need to know, the patient is already born. Their genetic mutation is already developed. Is it reversible? You may never be able to reverse it because the human baby is already born. They're 3 years old, they're diagnosed. So everything's formed. Can you reverse that? Right? So we think every gene is different because it depends on gene expression regions and expression time. So we tested this idea. And two former graduate students, Karen May and Patricia Montero, did this experiment. Patricia is a group leader now in Portugal, University of Minnesota, and Karen is a postdoc in San Diego, UCSD. So we decided we developed a new mouse line, allow us the mouse to grow up like a knockout, a mutant mouse. They have all the phenotype, gloomy, their skin off, social interaction deficit, motor problems, anxiety. Then this mouse can be crossed to another mouse that allow us to re-express by giving a drug called tamoxifen and induce the expression of the normal copy of the SHANK3. So now you can feed this drug anytime, right? In adult, in 20, a month-old mouse, a week-old mouse, to see when is this reversible? If it's reversible, when it's reversible. That gives us some idea. Is there a hope to develop gene therapy for this type of monogenic disorders? And so what we did exactly what I said. We let the mouse grow up into 3 or 4 months old. They show all the phenotype, including grooming their skin off. Then we give them tamoxifen, turn the SHANK3 gene back on. Now you suddenly, these mice never see a SHANK3 gene, a protein, suddenly SHANK3 protein is available. So what happens in this adult, even in the adult, you can rescue molecular defect, you can rescue functional defect, and you can rescue even structural defect. That was a surprising. Actually, synapse is lost, but they can regrow back. So that is suggesting at least in some brain region, there is a plasticity in adult animal, right? So, and behaviorally is similar. So I mentioned that these mice will repeatedly grooming their skin off. So then at this time, you say, this is the same mouse, you turned SHANK3 gene on in adult. Suddenly, after, this is after 2 months, you actually see that the skin heals and hair grow back. The only difference is the hair grow back white. This has something to do with tamoxifen. Actually, tamoxifen is a breast cancer drug. In patients, you take tamoxifen, some patients also hair turn gray. So suggesting that in at least this behavior, not only you can functionally reverse it, and the behavior you can also reverse it. Similar thing happens to the social interaction deficit. We use called a 3-chamber assay to assay the mouse social behavior. In this case, there are 3 chambers, left, middle, and right. You put a mouse in the middle, and there's a little cage here with a stranger mouse, a little cage here with an object, and the mice can freely go to either side. And the wild type of animals like to social with stay around with this stranger mouse. Mutant animal actually didn't have much preference, but if you re-express SHANK3 in adult, now you restore this normal social behavior. Suggesting both repetitive behavior and the social interaction deficit can be reversed in adult. Now the question is, can we reverse everything? Is it actually turned out to be not? So many of the things, for example, reduce the motor activity, like hypertonia in patient. In this case, we use motor activity, anxiety, motor learning, and sensory hypersensitivity. All these, none of them are reversible in adult. So suggesting there's a limited adult plasticity. However, if we turn the SHANK3 back gene on earlier, still postnatally, for example, 21 days after birth, you can improve all these behavior. So this suggests that at least for the SHANK3 mouse model, that they are indeed adult, there is an adult plasticity in some structure and function of the brain. However, there are also critical developmental windows for other circuits and behavior. This is very consistent with the critical period, right? So language and cataracts, a vision system. So they are indeed a critical period. But at least from this study, there's a hope that there is a postnatal window for gene therapy. That means if you can replace the gene, of course, our previous study is a genetics. You cannot go on to do a genetics in humans, but we can develop a gene therapy for use other approaches to see whether you can postnatally, you can rescue this behavior. So we did exactly that. So currently, the most effective way for getting the genes into the brain is the use called adeno-associated virus, AAVs. This is very safe, as there are hundreds of clinical trials going on for a variety of different disorders. So, but AAV has an issue, is that it can only packaging a very limited amount of DNA into it. And many or most of our brain genes are very large. So SHANK3 is way larger than the AAV can package. So we actually developed, my graduate student actually developed we call it mini shank. Basically, we look at the function, structure and function, and delete the region we think is not critical for its function. And we made it much smaller so it can be packed in AAV so we can deliver into the brain to do this experiment. And so what we did was to package this gene therapy construct into AAV, then systemically administer to P7, P4, postnatal day 7, postnatal day 14, postnatal day 28, or adult. You can test them. When can you rescue this, right? And you can see in mice, we tagged this gene with GLP, so you can see their expression. You can see they express really well. Then you can do, after 2 months of waiting, right, they become adult, you can test the molecular, behavior, and electrophysiological to look at what can you rescue and what can you basically we call the cure in mice. What we found is this construct can rescue all the defects from molecular, functional, and the behavior. Right? I'm just showing 2 points. One is the synaptic transmission from corticostriatal synaptic transmission. Black is the Y type injected with AAV GLP just as a control. The red is the mutant animal injected with AAV with GLP as a control again, and the blue is the the mutant animal injected with mini-SHANK3 tagged with GLP. You can see functionally and behavior the same. This is social interaction deficit, right? So Y type like to social with stranger mouse. Mutant did not have a preference, but after you do the gene therapy, they restore like Y type. So this mostly work was by a postdoc now, Shan Gao, that did this work. So this suggests that indeed, there might be a postnatal window for gene therapy in patients. So now this contract actually, biotech company pick it up, want to test in humans in the next few years. And so we're hoping that, I met many of the parents with SHANK3 kids, mutant kids, SHANK3 mutation kids. They are desperate for some kind of treatment because they literally have to take care of them. It's a tragic case for patients, but it's also a tragic case for families. So this kind of gene therapy actually is now you can see a lot more, right? So this was just published, I think, last week. A group in UCSF showed that this is a different gene called AADC, and they locally inject into the substantia nigra to restore the function of the dopaminergic neurons. These are in patients. I think 5 or 6 patients. Actually, they are partially restored. So it's functional. So you will see a lot of this AAV-mediated gene therapy and for monogenic disorders. I think I'm very hopeful, and many of the parents and the patients are very hopeful. This will really benefit the patient. And now, this actually, we call them, this gene therapy, we call them gene replacement. You basically use a gene to replace the loss of function genes, right? But it has advantage, but it also has disadvantage, right? So first of all, some are too big. Not every gene can miniaturize. For example, if you have a sodium channel mutation, like Dravet syndrome, SN2A, it's huge. It's a membrane protein. It's a sodium channel. You cannot just miniaturize it because it's needed pore areas and need all these structures. And it's a huge, much bigger than SHANK3. So it has also you may worry about the overexpression. In some cases, overexpression is also a problem. So now the new genome technology called genome editing technology, you probably heard a lot. The CRISPR technology made this possible to precisely correct the mutation without overexpression, without put the gene back. Whatever the mutation you have already happening in your genome, I'm going to correct you without put another gene in, whether it's a mini gene or full gene. So this is you very familiar, right? Professor Gardner and Charpentier won Nobel Prize. My colleague and friend Feng at McGill Institute also made a great contribution to realize to develop this technology. And this is we think there are 2 approaches that are most useful. One is the correct gene. The other is regulated gene, increased gene regulation, because almost 90% of mutations are we call it haploinsufficiency. Most of genes, we have 2 copies, right? Except on the sex chromosomes. So usually mutation only 1 copy. The other copy is normal. It's just not enough. Well, if it's not enough, why don't we just go on regularly, make the other copy make more, then you can compensate for it. So these are the 2 technologies that are most promising in the near future. So the CRISPR, you're probably very familiar, right? So you basically this was used by bacteria to fight off virus, bacteriophage, right? So first time they got a bacteriophage infection, the bacteria will recognize the DNA, right? So it incorporate into genome, then it recognize Next time, same bacteriophage come, it will make RNA from its bacterial DNA, so it can recognize the phage. Then this called guide RNA can guide a Cas9 and nuclease to cut the phage. Now it will basically destroy the virus, so the bacteria can survive. So it's a defense system. So now we can use this for mammalian system. Basically, whatever gene you want, you can design a guide RNA, right? So we all know all the sequences, whether it's humans or mouse or Drosophila. And then you basically predict where it will cut based on your guide RNA. Once it's cut, our cell want to repair it. But the cell repair is sometimes not precise. So you need a genetic mutation. This way you can generate animal models. Then the other way is if you provide a template, the cell will replace with your template. So now you can correct a mutation or generate a new mutation. So these are the most common ways. So the advantage of CRISPR is its versatility, right? There's a reason we all use mice. When I started my postdoc, I wanted a mammalian system I can do genetics. That time it was only to do genetic engineering. That time it was the only mouse was the only species. And because we have developed ES cells and the technology. But CRISPR can be anywhere. So it can be can do gene deletions, repairs, and transcription regulations, but it also can be almost any species, right? So in animals, plants, bacteria, humans. And it also can do multiple genes. When I was supposed to knock out a gene, a mutated gene was a very, very long process. Now you can do multiple genes in just one, in a mouse. So this made it an incredible, powerful technology, right? So the most useful thing, I think, is not the original CRISPR-Cas9 system. Actually, it's the modified one. For example, David Liu at the Broad Institute developed called base editing. Basically, you inactivate the nucleus. You don't want to cut your DNA. It's too dangerous, right? So you inactivate the nucleus, but it still can bind to the sequence. Then you attach another enzyme, in this case, cytidine deaminase, to the Cas9. Now you bring this deaminase to the DNA sequence you want to modify. So in this case, you can edit a single base. For example, you want to change the GC to AT. This deaminase will deaminate the C and become a U. U in the DNA, it will translate into T. So now suddenly a GC is changed to AT. So you can make a mutation like this, or you can basically correct a mutation like that. So this is going to be tremendously helpful because about 80 or above 80% of mutation found in human are single nucleotide variations, SNVs, right? So this will be incredible technology. So this was just done by David Liu's lab in mice, show that base editing can in the stem cells actually can rescue sickle cell disease, right? So there are a lot of hopes for this kind of technology. So now the problem is every gene therapy development is very, very expensive, especially if you have to make a lot of AAV to put it into the human body, right? Current estimation costs about $1 million to make enough clinical quality AAV for treat a human. So then there's ethical issues. Who is accessible? We can access this, and how can we reduce the affordability? For example, sickle cells, many of them, most of them are in Africa. Some countries are really poor. And you have the technology, you completely can do it. You can save lives, but they cannot pay it. So as a society, as a whole, international community, we should think, how can we make them available to them, right? So these are kind of ethical issues. Current graduate student, postdoc, young people are very, actively involved, engaged. I always encourage them to think about how can we make it accessible to all the people around the world. Then another use, which is really reality for almost anyone, right? So, when we get old or we have a disease, our organ will fail. Getting a match is a really, really difficult thing. And sometimes you cannot get a match. So now many labs and companies start to, basically we call it humanize pig organs, right? With CRISPR technology, you can eliminate the genes or change genes that match, with tolerable or compatible to human. So there are companies, especially like George Church's lab, have done this, and they actually have a couple of companies now. So this is completely possible, right? So in the future, maybe each of us have 10 pigs, 2645 is Guoping Feng's pig, right? It matches my compatibility. So when I getting old, my kidney fail, I get a pig kidney and replace it. So in the future, many of us maybe just, yes, we live 100 or 120 years old, but all your organs are pig organs, except for your head is still your head, right? That's all matters. So you will see a lot of walking pig around in the near future. So this is not a science fiction, it's a reality, right? So it's just time, maybe 10 years, maybe 15 years. So this is come to another extent. If you have resource, you may live for much, much longer, and your life will be much better because whatever failure organ, you can get a replacement with your own labeled pig, right? Give it a number and barcode them. So these are all ethical issues. It's really will become a reality. I'm very confident it will become reality. Is who can access, how can we make it available to the people who really need them. So these are gene therapy and gene manipulations in somatic cells. That means in your cell as in your body, not in your germ cell like sperm or eggs. So that means it's not transmitted to the next generation. A bigger issue is when you change the genes in the germ cell. That means every single cell is changed. That's the best way to correct the mutation. However, you change the human race, basically, right? There are a lot of issues that can come. So my lab recently, just recently published, done by two research scientists, Jonathan Wild and Tomomi Ada, we will try to increase the ability to efficiency to generate, to manipulate genes in mouse embryos, so allow us to generate disease models more efficiently. And we externally identify method technology, actually develop a technology, allow us to very efficiently to change genes in embryos and without any template. For example, if you have a heterozygous, you have one mutation, another is normal, you can just use our technology, go into embryo, correct the mutation with this existing Y type. As I said, most of our heterozygous, they have one normal copy, one mutant copy, we just go in, use the Y-type copy as a template to correct the mutant copy. So this technology is very powerful because you can generate homozygous mutants, right? Instead of you make it normal, you can also make it, both are mutant copies, right? So, and now this comes the issue. We know there are many protective genes in our body. For example, Alzheimer's disease, right? When you reach 85, about 40 to 50% of us will have Alzheimer's disease. This is the largest scale human population studies. But if you have an ApoE2 homozygous, the chances are much, much, much lower you will get Alzheimer's disease. If you have ApoE4 homozygous, you have 40 times more, 20 to 40 times more likely to get Alzheimer's disease. So now with this technology, you can actually make them homozygous for ApoE2. Then you don't have to worry about it when you're getting old. And are we allowed to do that, right? So you can do very easily, and it will be in your family because it will pass along. So this is, ethical concerns, right? So now, maybe treating a disease is definitely acceptable. At least MIT Tech Review did a survey that most people, at least half of people, this is about 6 or 7 years ago now, and support this use to reduce the risk of serious disease by manipulating genes in embryos, in germ cells. And but if we're talking about, well, I want my kids smarter, right? Go to better schools, have better jobs, and most of people, at least so far, against that, right? There are good reasons. So let's say in intelligence, right? Intelligence is a bell curve, low performance on this side, but most of people want to be on this side. We cannot be Einstein. Einstein is outside of the bell curve. I don't know where that is, but we cannot create an Einstein. But they are, with large-scale genetic studies, pretty soon we will identify genes that correlate with IQ, right? So just we can correct intellectual disability. That means that gene is important for intelligence. Then we can also potentially in the future identify genes are protective or enhancers. That means you may move your family's IQ from this all normal, right? So we're all normal. It's just a bell curve from bell curve this side to other side. Are we allowed to do that? So all these ethical issues, I think the whole community, should engage and discuss. Just like in the 60s and 70s when we have DNA technology, and there was a huge debate and worry. At the end, there are standards established, and the medical science benefit a lot from DNA technology, especially the cancer field right now, right? So the brain disorder now we are starting to benefit. So right now, most scientists, including myself, are against embryonic germline manipulations because we don't know what else will happen. You are basically changing the human race, right? The DNA will pass along and passing down generations. So we don't know what the side effect, because you think very detailed, but nature sometimes do a lot of unexpected things. So I think a lot more research need to be done before we can actually go on that road. And I don't know whether they will ever, we should go to that road. So this is the kind of gene therapy I want to get an overview, but in the next, I just want to spend a few minutes to show, if they are not monogenic, how can we do it, right? So we can modulate the circuit function for polygenic causes. What are the technology now we can do that? So I want to mention, this is not new, right? Stimulated circuits to treat a disease has been used for decades, right? The most famous is the deep brain stimulation, put an electrode in the brain, and in Parkinson's disease, they're actually quite effective, right? The disadvantage in this older technology is you need a large electrode into your brain. So and then you have battery outside, you have stimulator outside. There are many complications. It's a large surgery, and also infections, scars, and inflammations, all these issues. And also, it's not a precise stimulation. Actually, you see this electrode have multiple spots. You basically test which spot is better. And once you implant it, okay, maybe this spot is better than this spot is ineffective, then you can choose to stimulate this spot, right? So it's not a cell type of specific stimulation. And now the new technology can change that, right? So when you are very familiar, it's called optogenetic stimulations, that you can precisely express a protein, light sensitive channels, right, or pumps into a particular cell type, then you can very precisely stimulate that circuit, and you have much more effective and way less side effect. And the other is what we call chemogenetics, which is you inject, express a ligand-gated ion channel or receptors into particular cell type, then you just take a pill. That pill does not treat your disease. The pill is just activate this channel or receptors that can activate your circuits or suppress your circuits and treat the disorder, right? So the major, optogenesis already used in clinic. This is just published a week ago, right? So Banta Roska's lab in clinic, in one patient, they put the opsin, we call it optogenetics, with opsin into the retina cell. But these are blind people, they don't have photoreceptors. Usually your photoreceptor sense the light. Then it transduces a signal to the retinal ganglion cell, to neurons, then go into the brain, you can see. But in these patients, they are photoreceptor degenerated. So, if you put these light-sensitive channels into the retinal ganglion cell, they usually don't sense, now they suddenly can sense light. So that way you can restore certain vision. So this is already in clinical, this is in one patient, right? So it will be in clinic. But if you want to do in brain circuits, the problem is you need to put an optical fiber into the brain, inside the brain. So that's almost like deep brain stimulation. So it will cause damage to the brain, inflammations, scar tissues, all these things. So we actually decide to see whether we can improve the sensitivity of this optogenetic, and without put optical fiber into the brain, we can still activate precise the cell types and the circuits. So we developed a new opsin we call a SOL, a step function opsin with ultra light sensitivity. And so use this new opsin, we can show that if you put if you express this is the thalamus, which is the middle part of the mouse brain. And you don't need to put optic fiber in. You put an electrode in to record it, and then you shed lights from outside the skull, right? And you can see it activates, right? The original option does not activate, but our soul activates. So now the question is, can you use that approach to change behavior? So we tested in the deepest part of the brain called hypothalamus. Hypothalamus control many behaviors, including feeding, right? Brad Lower's lab at Harvard showed that if you put optic fiber in, then express channel rhodopsin, opsins here, you activate it, it will suppress feeding. In our case, we don't need to put optic fiber in. We just express this soul into here, use AAV again, express here. Then you share the lights from outside. Can you suppress the feeding? So this is what happened. You turn lights on, control still feeding, right? But the mouse expresses soul. You turn lights on outside the skull, you suppress feeding. You turn the lights off, mouse started eating again. So now you can actually remotely, without the optic fiber even inside the skull, can do that. This was done by a former graduate student, Shane Gong, and in my lab, and a postdoc fellow, Diego Mendoza Holiday, in Bob Desmond's lab. So now you can potentially do this. The issue, of course, the human brain is much deeper, much bigger, and skulls are much thicker. So we actually show that in macaque monkeys, skull is also much thicker. We can put optic fiber inside the skull without touch the dura or the brain. You can activate neurons in the cortex, throughout the cortex. So now, if you look at the tissue, you put it off, this is a staining for activated microglia. If you stain tissue, you can see it's inflammation in the brain. But in the transcranial activation, no inflammation at all. This is almost identical to the non-treated or the contralateral non-treated no light. This is with light, right? So you can see this has a huge benefit for non-invasive optogenetics. So if this can be used in the future in the clinic, this will be a very easy way. So I want to stop here just saying, now if you think about what can you use for, this new generation optogenetics or chemogenetics, right? For depression, there are clinical studies identify regions are hyperactive, and if you suppress that hyperactivity in the CG25, and you become happy. Don't you want to do that? So then the question is, how depressed should I be, or could I add a little bit higher dose to make me happier? All these are become ethical issues. And this is not the only case for the deep brain stimulation already identified patient. They wouldn't take it away. It did not treat their OCD, but it made them happy. By IRB protocol, if you does not treat your condition, you should take them out. But the patient refuses, say, I have never been so happy. I don't want to take it out. But this will become much easier. You don't have to, you just inject the AAV into, take a pill every day. How much should I tell? How much? So, and it's the same thing for chronic pain. I have back pain. Don't you want to be able to take your phone out, press a button, and activate that light, and your pain is gone, right? So all these are possible. It's not a science fiction. It's all possible that you couldn't sleep. You time it on your cell phone. 11 p.m., I go to sleep. You put a head on, you activate and go to sleep. And you say, I was 6 o'clock, I want to light off, I'm going to wake up. All these are become a possibility in the future. So the question is, what is we should do, what we should not do, and what is ethical, what is not ethical. So all these questions are very complicated. I cannot answer at all, but I like to think it. I encourage my students to think about it. So I'm going to stop here. I mentioned all the people who did the main work, but I want to special thank all my members, and many of them, I cannot mention. It's a really pleasure, honor and to work actually with all and the people in my lab. They are very passionate. They are very dedicated. They understand what they do may seem small, but they contribute to a much larger cause of to the society. And I also want to thank all the funding agencies that help all the work without, especially from NIH, but many individuals and foundations, private funding to support the lab in my lab and my postdoc and graduate student. Thank you.
Thank you so much for
Thank
a
you.
really interesting and thought-provoking talk. And do we have a little time for questions? Maybe just a few. If you have a question, put up your hands. We'll pass you the.
Can you hear me?
Yes.
Hi, so I want to thank you for the exciting presentation on the promising potential of gene therapy and surrogate simulation for ASD treatment. So I have two questions for you tonight. So from some of my personal experiences working with children with autism, a really commonly used therapy for ASD is the ABA behavioral therapy. I wonder, have you observed for any gene environmental interactions in mice with SHANK3 mutation or in any other ASD mouse models? And do you think there are any values in doing behavioral therapy at all for kids? And where do you think the future and funding would go to for developing ASD treatment?
Yeah, it's a very good question. So there are two type of, we consider there are two type of conditions. One is very severe, and they are intellectual disability. All these very severe conditions, the environment always will have some influence, but their influence is much smaller in these very severe neurodevelopment disorders than high functioning autism. So we haven't not done gene environmental interactions, but we for SHANK3 we only did one experiment. House wild type and a mutant mouse together to see what happens. And when you test their behavior, they are getting closer. So we got excited. It turned out because the SHANK3 mice are so anxious, they drove the wild type behave more like a mutant instead of other way around. So that's only the experiment we do. But for a lot of your second question, the behavior therapy is effective in the mild cases. And is more effective in mild cases. I think it's still extremely helpful. It's probably based on this data, the most effective treatment right now. But in the future, if you could combine with other approaches people have been doing, is for example, open the critical period. All these things might facilitate and make the behavior treatment much more effective. That's the theory, not a practice yet. So.
Thank you for the wonderful talk, Dr. Feng. So I have a question about the last part of your talk about using light to activate hypothalamic neurons to suppress appetite. So how do you imagine actually, so first of all, how did you specifically direct specific hypothalamic neurons to suppress appetite? And second of all, how does that really affect day-to-day living in certain patients that actually have hypothalamic lesions or mutations? And do you imagine them wearing headsets that would have flashing lights at very millisecond timescales? Like, what do you think?
Yeah, so this is a really good question. So we use that as a test case, not actually for this condition, because that's the deepest part of the brain. There's a very clear readout. For the future, you know, with the thing I didn't have time to talk about. With the single cell RNA-seq technology, you can identify all the cell type, what they are, how they connect. So in this case, if really in the therapeutics, it's probably will be genetics. Basically, you have the gene, the DRADs, for example, the chemogenetic tools in expressing this, in these cells, a specific group of cells. Then you just take a pill. Right? You can dose control how much you want. So that actually is much more for the circuit control, for the cell type spec activation or inhibition and for therapeutic purpose, not the optogenetic. It's very deep in the human brain. I don't think you can get there without optic fiber, at least now.
Question, right? Okay.
There's evidence that you can delete certain autism-related genes specifically in the peripheral nervous system and cause autism-like behaviors. And I was just curious about your opinion on those studies and your opinion on the role of the peripheral nervous system in autism-like disorders, or excuse me, autism spectrum disorders.
Yeah, a very good question. So yes, there are some evidence that regulated the similar genes in the peripheral nervous system also contribute. I have no doubt that's true. because, for example, Shang-3 is expressed both in the central nervous system and the peripheral nervous system. I think both of them are in some cases, for example, sensory, it may play a more important role. But for social and other things, the central nervous system maybe play a more important role.
Guoping, thank you so much for just tremendously exciting talk. I'm sorry that in many years we would then go off for a lovely reception, but in 2021 this is not available. But we give you our thanks.
Thank you. Thank you.